Good morning, grade 11 students. In this session, we're going to start with genetics revision for week 25 for grade 11 sciences. So here we start with the first definition of the alleles. Alleles, they are divided into either a dominant allele or a recessive allele. Dominant allele is the one that is the strong allele. It is expressed usually in homozygous or in heterozygous. Homozygous, we said, is the pure, where we have two identical alleles. For example, we have two dominant alleles, or heterozygous, which is called the hybrid. We have dominant and with recessive. And sure, the dominant will overcome the recessive. هيتغلب عليه. So this is the case of the dominant allele. It is expressed in both homozygous or heterozygous, and usually it's symbolized by a capital letter. While the recessive allele is the weak allele. It cannot be expressed unless in the case of homozygous, in the pure, where two alleles they are recessive, so two weak alleles together, so this will lead to the expression of the recessive allele. So the recessive allele is expressed in both homozygous and recessive allele, sorry, it's only expressed in the homozygous. Dominant allele is expressed in both homozygous and heterozygous. Do you want me to know, Mr., that homozygous is also pure and heterozygous hybrid? Yes, because in the exam, maybe I'm going to give you one of these. I'm going to give you pure. If you don't know that pure is homozygous, or if you don't know that hybrid is heterozygous, this is a big problem. So here we have in this example, they gave B capital, it's for the brown dominant allele, and B small is for the blue allele. Some of you, they ask me, is necessary to give the symbol B for brown and the symbol also B for blue? I prefer not. You can do this. By the way, B capital, this is the B capital, and not this is B capital. So pay attention to this. B capital is this one. Okay? This is great kgs. B capital, B small. This is B small. قد ما كبرتها حتضلها B small. So this is the B capital. B small هي هيدي. Okay? So I prefer to give other symbol than B. So for example, if you give for the brown B capital, for the blue you can give the letter that you want, R, S, T, U, V, whatever. But you have to signate by symbols. So you have to let L capital be the symbol of the blue, let's say. Okay, so you have to give the symbols. Whenever you give the symbols, it will work. In the quiz, some of you, they did what? They worked the exercise and they did the factorial analysis and without mentioning. For example, they used the B for the brown, let's say, and they used the B small for the blue. First of all, whenever you did this and you mentioned this like this without saying let B capital be the symbol of the brown allele which is dominant over the blue recessive allele, and let B small be the symbol of the blue recessive allele with respect to the brown dominant allele, you have to mention them like this. If you said B arrow for the brown, B small for the blue, that's not accepted. This is first. Second, if you move without mentioning what is the B capital and what is the B small, I'm not going to predict what is B capital and what is B small. If you use this without mentioning the symbols, this means that the exercise, the whole exercise, the factorial analysis, and the other parts are going to be neglected. So from now on, pay attention to this note. From now on, if you don't mention the symbols, this means that these symbols are neglected. I will neglect them. So in the quiz, I have 2 to 3 papers. They didn't mention the symbols, and they work the whole exercise according to their symbols. Okay, you think that B capital is for the brown and B small for the blue. Okay, but I don't know. So next time, this will not be accepted. Okay, so this will not be accepted next time. So let's continue, please. Starting with the dominant and recessive. Here we have the codominant allele. Codominant allele is the one that is expressed in both the homozygous and heterozygous. Yes, codominant allele is the one that is expressed in the heterozygous only, and not in the homozygous or heterozygous. So codominant is the heterozygous. For example, here we have a red fish crossed with a blue fish. The result is we have a fish that is striated blue and red. So whenever you see the combination of two phenotypes, this means that this is the case of codominance, where both alleles are expressed. Both alleles will be dominant. So here, the red R capital, R capital. Should I have to give the symbol? Let R capital be the symbol of the red allele, which is in codominant with the blue allele. And let B capital be the symbol of the blue allele, which is in codominant with the red allele. Here, gametes, it gives here only R, 100%. And here it gives only B, 100%. So what is the genotype of this fish? It's RB. Some of you is going to ask me, can I write BR? It's the same? Yes, it's the same. No problem. So both of them, they are dominant, and this is the case of co-dominance. Okay? Moving now, we talk about the phenotype. Phenotype is the sum of all traits: tall, short, green, brown, red, pink, small, red color, brown color, blonde, brown, whatever. All of them, they are called the phenotypes. It's sum of the traits: blood group, gene of the level of thinking, and so on. While the genotype is the sum of alleles. For example, I have the genotype BB or BS or SB or A small, AO, and so on. So always, always, and always, the genotype will code for the phenotype. True breeding. Well, the meaning of true breeding, already we said about this. True breeding is a line of individuals where the parents and the descendants or the offspring, they have the same phenotype. For example, black mouse is crossed with a black mouse. Black male mouse is crossed with a black female mouse. All the descendants, they are black. So this is called a true breeding line, where the descendants, the offspring, they are the same as their parents. Well, the meaning of homogametic, homogametic is an individual which gives only one type of gametes. Example of it, the female ovum. It has XX, if you remember. So it gives only one type of gametes. While we have the heterogametic that gives more than one type of gamete. Example, the male, it has XY, it can give X or it can give Y. So it's called heterogametic. Hollandric gene, if you were the word Hollandric gene, the gene that's carried by the chromosome Y, the Y chromosome, bidominant and birecessive. Bidominant, what the meaning of bidominant? Bidominant, individuals that have two dominant phenotypes. This case, we're going to talk about this in details in grade 12. In the case of dihybrid, you have two differences in two traits. So here in the grade 11 till now, we're talking only about the monohybrid cross. Monohybrid, the parents are pure and they differ in only one trait. This is one type of the crosses, which is called the monohybrid cross. So bidominant individual, they are having two dominant phenotypes. For example, has blood group A, and they are normal regarding albinism. Normal regarding albinism is the dominant. Blood group A is also dominant. A and B, they are all, both of them, they are dominant. So this is the code of bidominant. Birecessive, the opposite, have two recessive traits. For example, blood group O and albino. So this is recessive, birecessive. Dihomozygous and diheterozygous. Dihomozygous, it's individual homozygous for both traits. For example, I'm homozygous for the blood group. So I have A capital, A capital. This is homozygous for blood group A. Albinism, I'm normal and I'm homozygous. So this is D capital, D capital. Diheterozygous for blood group A, this will be A capital, O small, and DA, for example, regarding albinism. So this is diheterozygous, and this one is dihomozygous. Okay? So this is dihomozygous and diheterozygous. Always, always, and always, if the descendant they have the same phenotype as their parents, so this line is a true breeding line. What does this mean, true breeding line? Meaning that the descendants or the offspring, they have the same phenotype as their parents. Transmission of the gene. Here we have an example that we did it before about the gray female mouse crossed with the white male mouse. So always whenever we start a cross, always the parents is going to be pure, unless we cross F1 with F1 later on. So here we cross the parents, they are pure. So gray female mouse with the white female mouse. All the descendants, they are gray, like one of the parents. So first question, they are going to ask you, what may you deduce from this cross? So you have to say, after crossing a gray female mouse with a white male mouse, and sure the parents they are pure, and they differ in only one trait. So here we get in the F1 gray mouse like one of the parents. So we can deduce that the gray allele, and pay attention to this word. Not the gray is dominant over the white. The gray allele is dominant over the white allele, which is recessive. Here is the first deduction. Deduce from the first cross that the parents are pure and they differ in only one trait. So we got in the F1 in the first generation, in the F1 generation, we got F1 100% or all of them they have gray. So they are gray like one of the parents. So first of all, we can deduce that the gray, not the gray color, the gray allele is dominant over the white allele, which is recessive. This is the first step. Second, I will ask you, what is the type of this cross? Here, the parents are pure. You have GG, and here they have WW. So WW, and here we have GG. Okay? The parents are pure, and they differ in only one trait. So what is the type of the cross? It's called monohybrid cross. Monohybrid cross is one of the type of the crosses. Monohybrid cross, parents are pure and they differ in only one trait. What is the difference here? What is the trait? The trait here is the coat color, the color of the skin, for example, or the color. This is the trait. So here the parents are pure and they differ in only one trait. Mono, it's one. Monohybrid, they differ in only one. So monohybrid cross, parents are pure and they differ in only one trait. So we call it monohybrid cross. Then part 3, they were told you to give the symbols, designate by symbols. From now on, in the exam or in the quizzes, I won't ask you about the symbols. I will tell you what's not. For example, what can you deduce from this cross? What is the type of this cross? Then perform the essential factorial analysis to verify the results of F1, the طلعوا كلهم gray. So what you have to do is you have to give the symbols. So you have to say, let G capital, since it's the dominant and it's usually capitalized. So let G capital be the symbol of the gray dominant allele over the white recessive allele. Let W small be the symbol of the white allele, which is recessive with respect to the gray dominant allele. Here, I gave the symbol. Even though, I will repeat this and pay attention to this. Even though I didn't ask for the symbols in the exam or the quiz. Your job is to give the symbols. If you didn't give the symbols and you use them downward in the question, this question will be neglected, meaning that you gave symbols and you didn't explain for the teacher what are these symbols. لشو هيدول السymbols أو لمين. Okay, so this is your job. If I didn't ask for the part designated by symbols each of the corresponding alleles, you have to use them. Here you're going to ask me, Mr. Here you used the symbols before you said. Here I use them because they are mentioned in the figure here. Okay, so here I can't use them unless I gave the symbols. To do the factorial analysis, here I have to start to give the symbols and then to do the factorial analysis. F2, they had F1 crossed with F1, which gives the F2. F1 crossed with F1. This is another type of crosses. F1 crossed with F1. Here we call it the selfie cross. Selfie cross, whenever we cross F1 with F1. Okay, so here we have, till now we have two different types of crosses. The first one is called the monohybrid cross. The parents are pure and they differ in one trait. We call it monohybrid cross. Second, we call it the selfie cross, whenever we cross F1 with itself. So F1 cross with F1 is called the selfie cross. So if we want to move step by step, I told you what is the genotype of each of the parents and that of F1. The parents, they are pure and the gray is dominant. So sure it's going to be G capital, since they are pure. And here the white is recessive and the parents are pure. So W small, W small. Then gametes. How many different type of gametes here? They give only one type of gamete, which is G. Okay, and you have to put it in the symbol of the female. G, it's 100%. And here it gives only W, so you put it in the symbol of the male. and you put small w, and it's 100%. So here, the F1, they will be G capital, W small, 100%. So all of them, they are gray. And this explains that the gray is dominant over the white. Crossing F1 with F1, F1 with F1, so GW is going to be crossed with GW. So in the male, we have a GW, and in the female also, we have a GW. So we have in the male G 50% and W 50%. How did we get G 50% and W 50%? We have two letters. What is the chance to pick G? Make it like this, it's simple. So what is the chance to pick G? I have two letters, G and W. 50% to pick G and 50% to pick W. Also in the male, 50% to pick G and 50% to pick W. So here we make it like math, expand. So G and G, it will be GG. How did we get 25? 50 times 50 divided by 100. Or simply, you can cross the zero here and you cross the zero here. Sure, crossing in your mind, not on the paper. So you cross 50, 50, cross 0 and 0, you multiply 5 times 5, it's 25. Second, G with W, it's GW, so it's 50 times 50 over 100, so it's 25. And here is GW 25 and WW is 25. So I want to add GG will give gray. GW also will give gray as well as this GW. So we have 50% GW, while the rest is white, so it's 25%. If I told you, pay attention to this question. If I told you what is the risk for having what is the proportion for having a white mouse? Who can tell me what is the proportion of having a white mouse after crossing F1 with F1? Raising your hands, please. What is the proportion for having a white mouse? Yes, Lynn.
1/4.
1/4, very good. Sometimes I'm going to ask you not what is the proportion, what is the risk, or what is the probability to have a, for example, a white mouse. It's 1/4, very good. Now, if I ask you, Yahani, what is the risk or what is the probability to have a gray mouse, which is a heterozygous? Pay attention to these questions, they are very important. Heterozygous and the gray, what is the chance?
Proportion?
Yes.
It's 1/2.
1/2. How did you get 1/2?
Kemal, 50%.
50%, very good. So here you added 25, 25.
Plus 24.
So it's 50%, so it's 1/2. Very good. Sometimes I ask you, I want it gray and it's homozygous. What do you think, Nada? If it's gray and it's homozygous, what is the proportion for this? Gray and homozygous. Yes, Nada? Nada's here, right? Yes, Nada, do you hear me? Uh-oh. You can said instead of Nada, and I need other than Lean and Honey. I need a new voices. I want to hear.
Is this
Yes, Nada. So, what is the proportion to have a gray mouse that is homozygous?
1?
1. Gray and homozygous. Gray and homozygous. What is the homozygous? If it's a gray and homozygous, what is the genotype? What should be the genotype?
G.
Only GG. In the table of cross, where do we have GG? It's only here. Right? So it's not 1. It's 1/4, 25. So it's 1/4. Okay? If I told you, yeah, Mahdi, what is the proportion to have a gray mouse? Only gray, yeah, Mahdi.
1/2.
1/2? How did he get it 1/2? Yes, Mahdi. How did you get it 1/2?
White provides and 1/4.
Okay, I need a gray. I mean a gray. What is the proportion to have a gray? Not white. ما بدي white. Gray.
3/4.
3/4. Why? Here we have GG 25 25 to 25, so it's 75. If it's in proportion, so it's a 3 over 4. Okay? So the proportion is a 3 over 4. Thank you, Mahdi. Thank you, Nada. So let's move now. So first, after crossing a female gray mouse with a male white mouse, we get a homozygous F1. So you have to get familiar with this way, how to write this. So they have a gray coat. So the character gray color is dominant over the character white color. So you can say here, instead of the character, you can say the gray color allele is dominant over the white color allele, which is recessive. And here I gave the symbols. Okay? So homozygous and heterozygous is for genotype, homogeneous, heterogeneous for the phenotype. Steps of the factorial analysis. Let's remember these steps. First step, you have to write the phenotype of the individuals. Second step, you have to write the genotype of the individuals. Third step, you have to write the gametes. Fourth, table of cross, which is called the Punnett square. And 5, the table of results. So here, parents. First, I started with the phenotype of the parents. So phenotype of the parents, male and female, I have to give the symbols. So here the female, they are gray, and the male, they are white. Genotype, GG crossed with WW. And here we have gametes 100%, and sure you have to put it inside the symbol of the female, and here you have to put it inside the symbol of the male. And 100% they have GW, and they have 100% gray. If I ask you to verify the results, you have to say at the end, therefore, the results are verified. Here, crossing F1 with F1. Crossing the F1 with F1, we have GW crossed with GW. So G 50%, W 50%, as well as in the female, GW, G 50%, and W 50%. In the table of cross, in the table of result, we get 75% gray, 25% they are white. Moving to the tested cross. Why do we do the tested cross? Tested cross is the third type of crosses. So our type can the monohybrid cross, when the parents are pure and they differ in only one trait, we call it the monohybrid cross. Second, I have the selfic cross. Whenever we cross F1 with F1, it's called the selfic cross. Third type is the tested cross. Pay attention to this tested cross. Tested cross, it's always done between organisms whose genotype for a trait is unknown. So we have an unknown genotype for individual, and this is crossed with an organism that is homozygous recessive. Always. For example, let's say we have a gray and we have a blue. And in this exercise, we have, for example, white and we have a black. Okay? So always the white is recessive. So always it should be B small, B small. It's recessive and it can't be expressed unless in the case of homozygous. So it's sure it's B small, B small. The big question here is this black. It is B capital, black. They gave the symbols here. Or it's B capital, B small. It's heterozygous. So in order to know this, if it's the homozygous or heterozygous, so you have to do the tested cross. Tested cross, if we get two different phenotypes, meaning that here we have two different types of gametes. So always it depends on the unknown. Since the known, it only gives one type of gamete. It gives only B, 100% if you want. But the heterozygous, it gives two different types. So if you have two different phenotypes, you have 50% black and 50% white. So the unknown should be heterozygous. Why? Because they have two different types of gametes. Whatever, if it's homozygous, it gives only one type of gamete. So one type of gamete, so it is one phenotype. So أنا دايما, whenever I have a tested cross, whenever they cross with homozygous recessive, the first I have to know this is a homozygous recessive, it's a tested cross. This is the first. Second, I have to look at the results. If we have two different phenotypes, so the unknown is heterozygous. Why? Because it will transmit two different gametes. However, if we have only one phenotype, so it is homozygous. Moving now to the case of intermediate dominance and codominance. Codominance, we said that both they are expressed. Both allele, they are expressed, and both of them, they are dominant. So example about this, the A allele of the blood group and the B allele. A and B, they are dominant. Whenever you have an individual having blood group AB, sure, there is no blood, there is no one allele is called AB. There is allele A and allele B. So we have allele blood group AB. Both of them are dominant and both of them they are expressed. So this is the case of codominance. Or we can say, for example, cross a dog with the white color with another dog having a black. So the dog, their offspring, they become black spotted with the white spots. So here, both alleles, they are expressed. So this is cooperation between two alleles. So this is the case of codominance. However, in this figure, snapdragon plants, a cross is performed between red and white. The color in the F1 is in the middle between the red and the white. So this can be explained that we have an intermediate phenotype, which is the pink. So this is the case of incomplete dominance or intermediate dominance, where both alleles also they are dominant. For example, here the R is RR genotype, and the white is W capital W capital. Also, the pink, here we have R as a gamete, and here we have W as a gamete, so they will be R capital W capital. So if we cross R capital W capital, we'll have 20% will have RR, 50% will have RW, and 25% will have WW. So here, this is the case of intermediate dominance. And this is similar to the one that we solved in the quiz. We have a long radish crossed with round radish. The result we have an oval. We have an oval. So this is the case of the intermediate dominance where we have an intermediate phenotype. So here, let's move to the types of crosses. I said about the type of crosses. I said we have first, and pay attention to this. The first one is called the monohybrid cross. So this is the first one, monohybrid cross. One, the parents are pure and they differ in only one trait. So this is called monohybrid cross. Parents, they are pure. What the meaning of pure? They are homozygous. Parents are pure and they differ in only one trait. Already we talked about this. And differ in only one trait. Differ in only one trait. One trait meaning that when only one characteristic. For example, in one trait, it differ in only the color of the hair, the color of the eyes, the color of the skin, say for example the type of the ears, whether it's attached or freeload, and so on. This is a monohybrid cross. Second cross, we have self-cross. Upon crossing two F1 individual self-cross, it's F1 crossed with the F1. This is called the self-cross. F1 crossed with F1. Third type of the crosses is called the test cross. Test cross, whenever we cross an unknown with homozygous and recessive. So this is called the test cross. These are the three different types of crosses till now: monohybrid, self-cross, test cross. Moving on to the ways of transmission. Way of transmission of the hereditary traits. So here we have first, if we have a dominant and recessive, this is the case of dominance. We have one dominant and one recessive, like gray and the black, for example. All of them, the gray and white, all of them, they are gray. So this is the case of dominance. Second, we have the case of codominance, where both alleles, they are expressed. Okay, we have cooperation of the alleles. Third case, which is what? Number three, third case, which is the case of intermediate dominance or incomplete dominance. Okay, so this is the intermediate dominance or incomplete dominance. Okay, intermediate or incomplete, they are the same. Incomplete dominance or intermediate dominance, or you can say or incomplete. So here we crossed a white with a red, the result they are pink. So it's a color which is intermediate between the red and the white. So this is called incomplete dominance. So these are the notes that you have to know them before we move to review about the karyotype, about the chromosomal formula, about the chromosomal abnormalities, anomalies, and so on. So these are the whole notes required for this session.